Hello and welcome to Mobile Topic 4 on Transcription and Translation. After Topic 4, you should be able to describe the gene transcription and translation processes. There are 8 learning outcomes in this topic. Describe the role of RNA polymerase and functions of promoters in transcription. Explain transcription in prokaryotes and its polycistronic nature. Explain initiation, elongation, and termination stages of transcription and translation. Explain universality and degeneracy of genetic code. Describe post-transcriptional modifications of RNA, RNA splicing, and editing in eukaryotes. Contrast the major differences in transcription between prokaryotes and eukaryotes. Explain the use of genetic code during initiation and termination processes. And finally, explain the major translation differences between prokaryotes and eukaryotes. Let's start with part 1 on transcription. There are 5 learning outcomes in this part, mainly focused on the use of RNA polymerases in transcription, as well as the entire process of transcription in both prokaryotic cells as well as in eukaryotic cells. You should be very familiar with this slide by now. We talked about the central dogma in the previous topic as well, when we're talking about DNA replication. Central dogma is the description of the flow of genetic information from DNA to protein. And the processes which are highlighted in blue in this slide are what we're going to describe to talk about today, which is the process between DNA and RNA. The process from DNA to RNA is called transcription, and the process of RNA to DNA is called reverse transcription. Transcription is a process of synthesizing RNA from DNA sequences, which access the genetic information in the form of RNA without altering the structure of DNA. On the other hand, for reverse transcription, it's a process of making a double-stranded DNA from a single-stranded RNA template through a reaction catalyzed by reverse transcriptase. Reverse transcriptases are characteristic of retroviruses, for example, HIV virus. We'll talk about reverse transcription in more detail in topic 5 when we talk about RT-PCR. Let's start with the transcription process in prokaryotes. Transcription is a 3-stage process. It starts with initiation, where RNA polymerase recognizes and binds to the promoter region of the DNA, thus causing the DNA to unwind to form an open complex. After initiation, the transcription moves on to elongation, which is the main process of the synthesis of the RNA transcript. During this process, RNA polymerase will slide along DNA template, which forms an open complex similar to the replication bubble in DNA replication. And in the process, it will synthesize RNA. And lastly, termination. Once the RNA polymerases reach the terminator region of the gene, the RNA polymerase and the mRNA transcript will dissociate from the DNA, thus terminating the transcription process. This is an overview of what transcription is like in prokaryotic cells. We're starting from initiation, where we have the RNA polymerase that binds to the promoter region of the DNA with the help of the sigma factor. And this binding of the RNA polymerase will form something called an open complex. Okay, and after the initiation process, RNA polymerase will glide along the DNA. As you can see, the DNA is actually unwound into in sort of two single strands. Okay, and RNA polymerase is going to actually glide along DNA, and in the process read the template strand of the DNA. And as it glides along, it will actually unwind the DNA as it travels. And as you can see in step 2, elongation or synthesis of the RNA transcript, we have this golden piece of mRNA transcript that's going to actually come out of the RNA polymerase. Okay. That is synthesized by the RNA polymerase by base pairing. And as you can see, the blue fragment, which is the sigma factor, dissociates from the RNA polymerase after it actually starts to move along the DNA. And of course, after the RNA polymerase actually reads the DNA, you will actually rewind back into the double helix behind the RNA polymerase, or upstream of the RNA polymerase, to revert back into a double-stranded structure. And lastly, when the RNA polymerase reaches the terminator region, which is the gray color region here, the termination process will actually occur that will actually release the RNA polymerase from DNA, and it will also release the completed mRNA transcript from the entire complex. Now, one thing that you have to remember is that RNA polymerase, very similar to DNA polymerase, also adds new nucleotides onto the 3' end of the growing RNA chain. Okay, so it's basically operating in the same direction as the DNA polymerase. This is another illustration of the transcription process. So at the beginning, we have the sigma factor together with the RNA polymerase that will actually bind together to the promoter region of the DNA. And then when the RNA polymerase move on onto the DNA, you can see the sigma factor gets dissociated from the complex, and the RNA polymerase is then able to generate the RNA transcript that it's supposed to generate. And you will move on all the way until it reaches something called the transcription terminator region on the DNA, and that's where the RNA polymerase as well as the newly synthesized RNA will be dissociated from DNA template. Okay, so let's take a deeper look into the 3 steps of transcription. We'll start from initiation. It is actually a recognition step where the promoter region of the DNA, which is upstream of the gene coding region, the protein coding region of the gene, will actually function as a recognition site for the sigma factor, which is a transcription initiation factor. Now, when after the sigma factor recognizes and binds to the DNA at the promoter region, it will then cause the RNA polymerase to also bind to the sigma factor as well as the DNA at the promoter region, and this would cause the DNA to start to unwind to form the open complex. At the initiation stage, RNA polymerase will glide along the DNA template. It will reach the region of the DNA. It's called the promoter region, which is going to be upstream of the sequence of DNA that's going to be transcribed. After the RNA polymerase is reach the promoter region and recognizes it, you'll bind to it and start the RNA synthesis. After the binding of the RNA polymerase, the DNA double helix will be opened up to form an open complex with the RNA polymerase, and that will expose a short stretch of nucleotides that can actually base pair with the new RNA that's going to be synthesized. Of the 2 DNA strands, only one of them is going to be acting as the template, and the RNA polymerase will start to incorporate ribonucleotides into the enzyme, which are going to be complementary to the DNA template strand, and that will in turn generate the new RNA strand. Now, one thing that's different between RNA polymerase and DNA polymerase is that it doesn't actually require a primer to start the synthesis of the RNA strand. In other words, it's actually able to um, attach new nucleotide without a free 3' end. And let's take a look at a couple of characteristics of transcription initiation, which are unique to prokaryotes. For prokaryotic cells, there's only one type of RNA polymerase that's around, and transcription start signals encoded in DNA are different between different species. There are also some consensus sequences. By consensus sequences, we mean some of the nucleotide sequences are actually common, and the transcription starts in all the promoter region. And why are they kind of some different? Why are there some differences there? It's because some of the sequences actually determine the strength of the promoter. So some of the promoters which are stronger will probably enable higher level of transcription of the particular genes. Okay? And of course, there are weaker promoters, which then usually precede the genes which are more rarely transcribed. For example, in E. coli, the promoter region usually consists of two important sequences, the -10 sequence and the -35 sequence. Okay. Both these sequences are actually upstream of the transcription initiation site. Hence, because they are promoters are always upstream of the transcription initiation site. The RNA polymerase will actually recognize these two sequences, and they will actually bind to these promoter sequences. And they will help to guide the polymerase to be at the right location to start the transcription. So you can imagine that after the RNA polymerase recognizes these two sequences, they will actually help to put RNA polymerase at the correct place where it can actually open up the DNA strands. Okay. And the DNA strands open up at the -10 region, which has a higher percentage of ANTs. This is actually often called Tata box for its higher population or higher probability of A's and T's. Okay, and this is the region where it's also a little bit easier to open up because of the fewer hydrogen bonds between A and T's as well. Moving on, the next step is elongation. During the elongation process, the sigma factor, which is this blue thing here, is released from the RNA polymerase, and the RNA polymerase is going to glide along the DNA in an open complex, meaning it will actually continuously open up the DNA double strands to be able so that it is able to read the template strand. The RNA polymerase will catalyze the linkage of RNA nucleotides, which are complementary to the template strand. So as you can see in this picture, as the RNA polymerase moves in a 3' to 5' direction along the DNA template, it will actually incorporate new nucleotides which are complementary to the DNA template in the 5' to 3' direction to synthesize the new RNA strands. And the RNA will have the same sequence as the coding strand of DNA, but as we learned in topic 2, there is actually no T in RNA, right? So all the T's will actually be replaced with U's instead. The RNA polymerases will glide along the DNA template in the 3' to 5' direction, and for each of the new nucleotides on the DNA strand that the RNA polymerase reads, you will add a complementary RNA nucleotide to the 3' end of the growing RNA strand. The RNA transcript that is generated is going to be complementary to the template strand that we're using. So in this picture below, it's actually showing you the two strands of DNA. One of them is called the template strand, and one of them is called the coding strand. The template strand is the strand that the RNA polymerase is actually reading So it's reading in the 3' to 5' direction towards the right. And the coding strand, as you can see, is actually going to be complementary to the template strand. And this is the strand that has the same nucleotide sequence as the RNA transcript that's generated. Of course, the difference is going to be at the use of nucleotides instead of T's. They're going to be used on the RNA transcript. And one thing that you may want to take note of is that the 5' end of the newly synthesized RNA transcript does not actually remain hydrogen bonded to the template strand after it's generated. Okay. And the template strand, which is the DNA strand, after being read by the RNA polymerase, is going to actually anneal back to the other DNA strand to form back the double-stranded DNA. And because of that, you can actually have multiple RNA polymerases which are reading the same DNA, so one by one, at the same time. So as you can see in this picture, the line in the middle, the darker colored line, is actually a piece of DNA. And as you can see, there's actually seemingly a cloud that surrounds it. And if you zoom in a little bit more, you'll notice that this cloud is actually a lot of RNA polymerases, a lot of RNA transcripts, which are actually irradiating from the DNA template. Okay, so on this piece of DNA, there are actually a lot of RNA polymerases which are reading it and generating transcripts out of it. So every RNA polymerase will actually have a tail, which is newly synthesized RNA transcript that's going to be trailing behind it. And as you can see in this picture, the cloud is actually a bit thinner on the right and a lot thicker on the left. That would actually indicate that the right side of the DNA is actually the starting point of RNA transcription. Okay, therefore, the tail that's irradiating from the RNA polymerase is a lot shorter. And when you move along the DNA, you notice that the RNA transcription on the left of the DNA are actually a lot longer. That means this is actually towards the end of the transcription region, the region of DNA to be transcribed. So how are genes transcribed on a chromosome? In the structure of any gene that's out there, they are always preceded by a promoter region, which are these lighter blue colored regions on the red chromosomal DNA. And the genes are always downstream of the promoter regions. So in this picture, there are actually three genes: gene A, gene B, and gene C. For gene A and gene B, the promoter regions are both on the left of the genes. So therefore, gene A and gene B are actually read from left to right, because the promoter region is always upstream of the gene that's being transcribed. Okay. So for gene A and gene B, the transcription actually takes place from left to right. And as we have learned earlier, the RNA polymerase is actually reading from 3' end to the 5' end of the DNA. Therefore, you can see that the bottom strands of both gene A and gene B are actually the template strands that the RNA polymerase is actually reading. And the top strand is therefore the coding strand of these two genes. Now, on the other hand, if you look at gene C, the promoter region of gene C is actually towards the right of this gene. Therefore, the transcription of gene C is actually going from right to left. And because of that, the top strand on the other, in this case, is going to be the template strand instead. So that's going to be the strand that the RNA polymerase is actually reading. and the RNA is going to have the same sequence as the bottom strand, which is also the coding strand of gene C. So depending on the location of the promoter relative to the gene, whether it's to the left or to the right of the gene, you should be able to decide which of the two strands of the DNA is going to be the template strand that the RNA polymerase reads, and which of the two strands is actually the coding strand, which is going to have the same sequence as the RNA transcript that's produced. Now in this slide is a little bit of a reinforcement on the complementarity between template strands of DNA and RNA, okay, where you can see that the A is always base pairing with T in DNA and U in RNA, okay, whereas G and C still remains. Okay, let's move on to the third and last step of transcription, which is termination. Transcription terminates when the RNA polymerase reaches the terminator region and is marked by the dissociation of RNA polymerase and the RNA transcript from the DNA. In prokaryotic cells, the termination of transcription happens in one of these two mechanisms: role independent and role dependent. In prokaryotes, there is no conserved termination sequences which the RNA polymerase always recognizes. So for role independent termination process, the mechanism of this process is basically stabilization of the transcription complex as it moves through the termination sequence of the DNA template. What happens during the role-independent process is that the RNA polymerase is going to encounter first a CG-rich region of the template DNA. Now, as you remember, if you encounter a CG-rich region, there is actually a stronger hydrogen bonding between the bases, right? So when you form when the RNA polymerase actually reads and synthesizes a CG-rich RNA piece, this will actually cause the RNA to fold back on itself to form a stable termination hairpin structure. Okay, so basically you can have a lot of the C's and G's going up to form a hairpin structure like this. Okay, so all these are actually CG pairs, and because they are CG pairs, this hairpin structure is actually very stable. And when that happens, the RNA polymerase is so now you have a very stable hairpin thing behind the RNA polymerase. And moving on, it then encounters another region of the template DNA, which is AT rich. Okay, so it's going to form another region here, which has very weak interaction with the template DNA. So now this hairpin structure here is actually going to cause the RNA polymerase to actually stop moving ahead. And because of all these very weak AT interactions, this becomes a point where you can actually break the RNA transcript from DNA template. Okay, and this instability for the enzyme will actually this instability that's happening here will actually cause the enzyme to break away from the DNA and also for the RNA transcript to be released into the cytosol. Okay, I want you to spend a couple of minutes to watch this video on role-independent termination. The other mechanism of prokaryotic transcription termination is called the role-dependent process. The role-dependent process is controlled by a protein called Rho, RHO, which is a helicase. Now, in the role-dependent process, the growing RNA transcript will actually contain the binding site for the Rho protein. Okay, so it's probably like about 3/4 down the actual RNA sequence. So the Rho protein will actually bind to that sequence, and you will actually move in the 5' to 3' direction along the growing transcript, and will follow behind the RNA polymerase. And when Rho protein is actually traveling along the RNA, it actually utilizes a lot of DNA. It actually utilizes a lot of ATP to help itself to actually move along the RNA. So near the end of the gene, the RNA polymerase will actually encounter a lot of G nucleotides. Okay, and this again will form a stem loop structure of the RNA transcript, and again similar to the role-independent structure, the role-independent procedure. This will cause the RNA polymerase to just stop at the terminator region. And when the RNA polymerase stops, the Rho protein can finally catch up to the RNA polymerase. And because it's actually a helicase, you remember that the Rho protein is actually a helicase, you will actually open up the RNA DNA hybrid, because it was actually they were tangling with each other. So this will actually help to open up the tangles, and this will also then cause the RNA transcript to be released from the DNA. Okay, I want you to spend a couple minutes again to take a look at this video and actually explain the role-dependent termination process. Now, in summary, this is what happens in prokaryotic transcription. There are 3 stages happening: initiation, elongation, termination. And remember the direction of transcription. The RNA transcript is always synthesized in the 5' to 3' direction, and that the RNA polymerase is always reading the RNA template in a 3' to 5' direction. Now, on the DNA, there is one strand called the coding strand, and the other strand called the template strand. The coding strand is going to have the same nucleotide sequence as the RNA transcript. The only difference is that all the T's on the coding strand are going to be replaced with are going to be replaced with U's. And the other strand on the DNA is called the template strand, which is what the RNA polymerase actually reads. Okay, let's take a look at what happens in eukaryotic cells. Now, as the eukaryotic cells actually have the cell nucleus, you can imagine that the transcription process actually takes place inside the cell nucleus. Now, the majority, or I would say the basic features of the transcription process in eukaryotic cells are actually very similar to the prokaryotes. We still have those 3 stages of transcription. However, because the eukaryotic cells are actually more complex, there are more cellular compartments, the transcription process is also more complex. Okay. So some of the complexity in transcription of eukaryotes comes from promoters, which are more complex. The more kinds of RNA polymerase that's present to transcribe different kinds of RNAs, and the use of transcription factors to initiate transcription. And very importantly, there are post-transcriptional modification of the RNA that's generated that helps to transport the RNA. Doesn't help, but those will actually assist in transporting the RNA from inside the cell nucleus to outside the cell nucleus, as well as ensuring that the RNAs that's transcribed remain stable in the cytosol. Okay, the first complexity that we set was the more complex promoters. In eukaryotic cells, there are a lot of regulatory elements and transcription factors which are involved in the initiation of transcription. In this picture, I'm showing you a eukaryotic gene. Okay. Now, the structural gene is the part that's going to get transcribed into the RNA. And upstream of the structural gene, we have a core promoter, okay, and we have something called a regulatory. We have a location where some of the regulatory elements can actually bind to. And these two things together are called the promoter region for the eukaryotic gene. So there are two elements in the eukaryotic promoter. We have the core promoter, which is the place where the RNA polymerase is going to bind to. And we have a regulatory element where some of the transcription factors will actually bind to either activate or deactivate the transcription, to stimulate or inhibit the transcription of that particular gene. Okay, so coming back, we're gonna take a look at the initiation process again. In this time round, it's about eukaryotic cells. For eukaryotic cells, the RNA polymerases are not able to actually initiate the transcription without the help of additional proteins called transcription factors. The transcription factors will first assemble at the promoters, and the promoters are again similar to prokaryotic cells, short sequences which are recognized by the transcription factors. And very importantly, I want you to always keep this in mind. The transcription factors bind to the promoters which are upstream of the transcription initiation site. And once the transcription factors binds to the promoter region, they will actually help to position the RNA polymerase at the polymerase binding site. For eukaryotic cells, there are a couple of different major categories of promoters. We have the Tata box, which is also very similar to prokaryotes, rich in T and A's. We have the GC box, rich in G and C's, which are usually found in housekeeping genes. And we have the CAT box, CAAT box, which are a little bit further away from the transcription start site, minus AT base pairs. So these are the three major categories of promoters which are present in eukaryotic cells. The second complexity of transcription in eukaryotic cells is that there are multiple types of RNA polymerases which are responsible for transcribing RNAs, producing RNAs in eukaryotic cells. So we're talking about three different RNA polymerases: RNA polymerase 1, polymerase 2, and polymerase 3. They are all responsible for different kinds of they are all responsible for the generation of different kinds of RNAs. RNA polymerase 1 is used exclusively to transcribe all genes encoding ribosomal RNA or RNA. We'll talk about ribosomal RNA in the next parts of this topic. And then we have RNA polymerase 2, which is transcribing all the structural genes or mRNAs. It also transcribes some of these small nuclear RNAs, which are needed for pre-mRNA splicing. And we have RNA polymerase 3, which are useful in transcribing all tRNAs or transfer RNAs and 5I and 5S ribosomal RNA genes. And we're going to focus mainly on RNA polymerase 2 because it's the main RNA polymerase that's transcribing the structural genes that we made into mRNA and subsequently made into proteins. Earlier on, we talked about the use of transcription factors in the initiation process. Now, for RNA polymerase 2, it actually requires 5 general transcription factors to actually help to locate the transcription start site. Okay, so these are proteins TF2A, B, C, D, A, B, D, E, F, and H. All these general transcription factors will actually assemble at the promoter region, which is a little bit downstream of the Tata box. Okay, and when they will actually assemble together with the RNA polymerase II at the transcription start site, and they will form a thing called pre-initiation complex. Only with the formation of a pre-initiation complex can the initiation process actually take place. Now we are skipping the elongation process because the elongation process is more or less very similar between the prokaryotes and eukaryotes. And the next difference in the transcription process comes in the termination process. Now for eukaryotic cells, unfortunately, we don't really know a lot about how the transcription actually terminates in the eukaryotic cells. One of the reasons is that there are just more kinds of RNA polymerases which are present inside eukaryotic cells. And every type of RNA polymerase actually terminates the transcription process differently. For RNA polymerase 1, termination of transcription takes place using a polymerase-specific termination factor that will actually bind to the DNA downstream of the transcription complex, and it will cause the transcription complex to store and to dissociate from the DNA. For RNA polymerase II, the process is a little bit more complicated. The RNA polymerase II is actually going to terminate around half a kilo base pair to 2 kilobases downstream of the end of the gene. Okay, so it's a little bit far from the end of the gene. But the mRNA transcript is actually cleaved at the internal site before the RNA polymerase II stops. Okay, and this will release the mRNA transcript. The next slide will actually show you sort of an animation of how the transcript is actually cut on the RNA by another protein. So the cleavage site usually is upstream of the termination site, okay, and it's usually quite rich in A, so it's usually an AA to AAA sequence. It's around 10 to 30 nucleotides down from the AAUAA sequence. The resulting mRNA transcript after the cleavage at the AAUAA sequence will be, of course, released from the DNA template, but it still has to undergo a couple of different modifications before it can actually be transported out of the cell nucleus. Okay. Now, one thing that's very interesting in this case is that even after the mRNA has been cleaved away, the RNA polymerase is actually going to stop transcribing. It's going to continue to transcribe along the DNA. Okay, but we will actually create a little bit of RNA that's actually not required, not necessarily that they're just wasted. So they will actually be removed by a 5' exon nuclease. And then once the exonuclease sort of catches up with the RNA polymerase II, it will actually disengage the RNA polymerase II from the DNA template, and this will actually terminate the transcription. Let's take a look at this animation. So in this picture, we have the red color thing is actually the DNA template, and we have a round RNA polymerase II that's actually reading along the DNA template, and yet we have this blue color RNA that's being generated. And as you can see, there's a region on the RNA that has the AAUAA sequence, and we have a cleavage site, the cleave site, which is a little bit after the AAUAA sequence. It's right before the GU rich sequence. And even after transcribing this sequence, the RNA polymerase II is going to continue to transcribe along the DNA and creating all these sequences that will eventually be digested away. As the RNA polymerase II continues to go ahead, the cleavage is going to take place. Okay, and this will actually cut away the pre-mRNA transcript. So this pre-mRNA transcript is now free to leave the site of transcription, and you will continue to undergo some post-transcriptional modification elsewhere. And we have this piece of the RNA still behind the RNA polymerase II, and we have this white color thing, which is the 5' exon nuclease, which is going to bind to the GU-rich region, and it's going to move towards the RNA polymerase II. Now, after it binds, after the binding of the 5' exon nuclease, it's going to actually move along the RNA, digest away the RNA, all the way until it reaches the RNA polymerase II. And from there, the RNA polymerase II is going to be dissociated from the template DNA, and this will terminate the entire transcription process. For RNA polymerase III, termination takes place with a series of U ribonucleotides on the RNA transcript, and U base pairing with A is actually quite weak, right? So this will cause RNA polymerase III to release the RNA transcript and dissociate from the DNA template. The next complexity of the eukaryotic transcription comes in the form of post-transcriptional modification, which is the processing of RNA after they are being made. So why do these RNA need to be processed? This is because eukaryotic mRNA is made as a larger molecule pre-mRNA, and it will actually be processed into smaller functional transcript. And one of the key reasons why it is so is because for eukaryotic genes, they are actually divided into exons and introns. So inside this picture, you can see that this is actually a structural gene, a eukaryotic structural gene. We are quite familiar with the promoter and terminator sequences already. There are also these red and green parts inside the gene, and they are called exons and introns. So what are they? Exons are things that actually will be present eventually inside the actual mRNA that gets translated, and introns are parts of the structural gene and the pre-mRNA that will actually be removed before the RNA is actually being used for translation. So you can think of an eukaryotic structural gene as being sort of discontinuous. So they are actually multiple parts. They're actually divided into multiple parts. So after a piece of mRNA is being produced, a piece of pre-mRNA is being produced. So remember, this is the part where the cleavage happens. Then there's like this AAU-AA sequence, so near the 3' end. This piece of mRNA is going to actually undergo a couple of post-transcriptional modifications. Capping, the addition of a 5' cap here. Okay. Splicing, which is actually removing the introns and sticking these yellow pieces, these exons together. And tailing, which is the addition of the 3' poly A tail. So as you can see, the mature mRNA has three features which the pre-mRNA doesn't have: the 5' cap, the removal of introns, and lastly, 3' tail. These are the three post-transcriptional modification that happens to eukaryotic RNAs, eukaryotic mRNAs. Let's start the process with capping. The cap is a 7-methylguanosine. It's a very special kind of molecule that is added to the 5' end of the eukaryotic mRNA. Now, this is actually happening at the very beginning of transcription process when the pre-mRNA is only about 20 to 25 nucleotides long, so while it's still being transcribed. Now, the addition of the 7-methyl-guanosine cap allows for the proper exit of the mRNA from the cell nucleus, and it's very, very important for the subsequent process that happens on mRNAs, which is the translation process, because ribosomes, eukaryotic ribosomes, actually recognize the 7-methyl-guanosine cap in order to bind to the mRNAs. Now, this is also important for the efficient splicing of introns later on in the post-transcription replication process. Next is the addition of poly-A tail. The poly-A tail is basically a string of A's, adenine nucleotides, which are added to the 3' end of the mRNA. This again occurs inside the cell nucleus after the pre-mRNA transcription has completed. So this is pretty much the region where we have the AAUAAA sequence, and then beyond that sequence we have that addition of poly-A tail. Poly-A tail can vary in length. It can be a few dozen A's, and it can be a couple hundred A's. So this will help to export the mRNA from the nucleus, and it also helps to increase the stability of the mRNAs and the lifespan of the mRNAs in the cytosol, so that they don't get degraded so easily. Now, before we talk about the splicing of introns, I want to briefly talk about how genes are actually organized in prokaryotic cells and in eukaryotic cells. Just to recap of the double-stranded DNA, we have one strand called the coding strand and the other one called the template strand. The template strand is the strand of DNA that's being read by the RNA polymerase, and the coding strand is the one that has the same sequence as the mRNA. So for if you look at the double-stranded DNA on the left, the top strand is the coding strand, and the bottom strand is the template strand. Where do you think the promoter region of this particular DNA molecule actually resides? Okay, in this case, the promoter region should be on the left of the double-stranded DNA, because the RNA polymerase actually reads this double-stranded DNA in the 3' to 5' direction. Okay, and the promoter has to be upstream of the double-stranded DNA. On the other hand, for the double-stranded DNA that's on the right, you can see that the red one, which is the coding strand, is actually at the bottom, and the template strand is actually on the top. So you can imagine the RNA polymerase reading this double-stranded DNA, the top strand of the double-stranded DNA from right to left, and therefore the promoter region for the double-stranded DNA on the right should actually be towards the right of that double-stranded DNA. We have also seen this picture a couple of slides ago. In this double-stranded DNA, again, we have this entire region is actually the gene that we're talking about. Okay, and there are regulatory sequences which are for the binding of regulatory proteins to influence the rate of transcription. And we have the promoter region upstream of the gene that signals the beginning of the transcription. We have the transcribe region that will eventually be in the sequence of the RNA. And lastly, we have the terminator region that signals the end of the transcription. Now, we have always been talking about the word gene, right? A gene is basically a DNA sequence that codes either the synthesis of a functional protein, in this case, the coding gene gets transcribed into DNA, transcribed into RNA, and then getting translated into protein. Or it can also be coding for a functional RNA, for example, RNA or tRNA. Okay, and these are essentially non-coding genes. So coding genes code for proteins, and non-coding genes don't code for proteins. The structure and organization of coding and non-coding regions of DNA very different. They are very different between prokaryotic cells and eukaryotic cells. So for bacteria, you have genes which are actually quite close to each other, with non-coding regions being very short. Okay, and then we have for yeast, Drosophila, and human, these are all eukaryotes. You can see that there are actually more spaces between genes, and even within genes, as you can see, we have like darker and lighter green colors, right? And the lighter green colors are essentially introns, darker ones are exons, so the darker colors are the parts of the gene that will eventually appear in protein in mature mRNAs. Okay, let's take a look at the difference in gene structures between prokaryotes and eukaryotes. Most prokaryotic genes have a relatively simple structure consisting of only three key regions: an upstream promoter region, a coding region, and a termination region. But one thing that's interesting about prokaryotic cells is that genes that encode proteins which function together usually, they will also be located together on the bacterial chromosomes. So the cluster of genes will actually share one promoter and one terminator. So you can imagine once the promoter is activated, multiple genes are going to be transcribed and translated eventually. So each of these clusters of genes is called an operon. And each operon can actually be transcribed to an mRNA transcript that encodes different proteins on one mRNA. And such an mRNA transcript is called a polycistronic mRNA. And genes in operons usually are co-regulated with other genes in the same operon because they are all regulated under the same promoter. This slide here shows you the typical structure of an operon. We have one promoter. So this operon is actually part of a prokaryotic chromosome, a circular chromosome. Okay, we have one promoter that's followed by three different genes, followed by a terminator. And of course, there is some untranslated regions between the promoter genes, amongst the genes, and between the gene and the terminator. This entire thing together is called an operon. So when you transcribe this operon, it will be transcribed into a piece of mRNA that codes for three different genes, that has three different genes on it, and they will eventually be translated into three different proteins, all controlled under one promoter. Therefore, they are called polycistronic. Now, on the other hand, for eukaryotic cells, the mRNA transcripts from eukaryotic cells are actually monocistronic, with one single mRNA that's encoding a single protein. However, eukaryotic genes are usually contain non-coding intervening sequences called introns. They are interspersed between the coding exon regions. And the vast majority of eukaryotic DNA is actually non-coding. Okay, so they don't actually code for any protein. For humans, only about 5% of DNA actually codes for proteins or functional RNA molecules. And this leads to our third post-transcriptional modification, which is splicing. Okay, the reason why we need to perform splicing is because we actually have introns found in eukaryotic genes, and they actually have to be removed during the transcription process. Splicing is a process of removal of introns and the joining of exons in pre-RNA transcripts. And there are two types of splicing mechanism that happens in the cell nucleus. Either self-splicing, meaning the mRNA will automatically splice out the introns, or spliceosome involved splicing, which is a more common type of splicing mRNA, which is transcribed from structural genes. And this is composed of several different subunits known as snRNP, small nuclear RNA, and protein. So remember, when we talk about the different populations of RNA, we mentioned snRNA, right? Small nuclear RNA. So they are, in fact, responsible for the spliceosome involved splicing when they actually interact with proteins and to actually cut the pre-mRNAs. Okay, so this is the structure of an eukaryotic gene. You see, this is a eukaryotic chromosome, which is linear. And when we look at this, when we take this out, we have one gene here that has a promoter region and multiple exons, which are divided by introns. When you transcribe this piece of DNA, you will get a piece of pre-mRNA, okay, that still has all the different introns inside the this piece of pre-mRNA. Now, remember earlier on we talked about the capping process and the addition of poly A tail. Now the next step is actually splicing, right? Post-transcriptional modification splicing. And this basically will actually remove the two introns from the mRNA and to actually paste the three pieces of of exons together. And this will be the full protein coding sequence that will be present inside the mRNA. And then this piece of mRNA will then be translated into protein. In eukaryotic cells, well, there's actually an interesting phenomenon called alternative splicing. So just now in the previous slide, we looked at how these three pieces of exons are actually pasted together to form the mature mRNA that looks like the first picture here. However, in eukaryotic cells, the connection of the three exons is not always happening in this way. So sometimes we can actually remove when we are trying to actually connect the exons together, exon 2 might actually get missing, and we have new variants of the mRNA that has exons 1 and 3 together. And similarly, we can have a new variant of mRNA that has exons 1 and 2 together, or you can have mRNA that has 2 and 3 together, or even have mRNAs that has only one exon. Okay, so through alternative splicing, even with just one gene, you can actually have multiple versions of proteins that comes out of one gene. So why is it important? There are certain proteins in eukaryotic cells that actually require different versions of it to function properly. One of them is actually our antibodies. Okay, so all antibodies actually are generated through alternative splicing of the gene so that we have different kinds of antibodies in our bodies. And with that, we finished our part 1 of the lecture on transcription. In the next part of the lecture, we're going to look at the translation process, how the mRNAs are being translated into proteins with the help of different populations of RNAs. Welcome back. In part 2 of this topic, we're going to be talking about translation. These are the 4 learning outcomes in this part: explaining the universality and degeneracy of the genetic code, explaining the use of genetic code during initiation and termination processes, explain initiation, elongation, and termination stages of translation, and lastly, explain the major translational differences between prokaryotes and eukaryotes. Again, we're coming back to the central dogma, and in this part of the lecture, we're going to be looking at the last process on the central dogma, which is translation. Translation is the process of translating the genetic information is written on the mRNA into working proteins. Let's start with the genetic code. The genetic code is basically the rules how the AUCGs on the mRNA can be translated into the language of the protein, which consists of 20 different amino acids. The genetic code is a set of rules that define how the 4-letter code of the genetic sequence, which in the case of mRNAs is A, U, C, and G, is translated into the 20-letter code of amino acids, which are building blocks of proteins. There are 5 different features of the genetic code that you have to keep in mind. The first one is that it's a triplet codon, which means every codon in the genetic code is comprised of a string of 3 consecutive ribonucleotides. So any codon that we're talking about always have 3 nucleotides. So you have things like AUG, or you can have maybe GCGs and things like that. So you can actually find on the table for the genetic code the corresponding amino acid, the amino acid that corresponds to a particular genetic code. The genetic code is also unambiguous. Every genetic code, every codon, specifies only a single amino acid. Okay, so the genetic code, for example, GCG, only can define one amino acid. However, the reverse of that is not true, which is the another feature of the genetic code called degenerate. Because there are only 20 different amino acids out there, but there are 64 possible genetic codes. So for any amino acid, there can actually be more than one codon that codes for this particular amino acid. So out of the 20 different amino acids, 18 of them can actually be defined by different codons. And only two amino acids are specified by one codon. Now, in this set of genetic codes, there are some very special codons called start and stop codons. The start and stop codons will tell the translation machinery where to start, to initiate, and to terminate, to stop the translation process. The start codon codes for methionine, which is one of the 20 amino acids which is coded by the genetic sequence. Whereas the stop codon, on the other hand, does not code for any amino acid. Lastly, the genetic code is nearly universal. The vast majority of organisms actually use the same genetic code when it comes to the translation process, be it viruses or bacteria or even eukaryotes. Okay, so what you're seeing right now is the genetic code table. You don't have to remember this for the tests or any exams because this will be provided to you. Now, the way to read the genetic code table is to start from the left. You see this UCAG in purple on the left. This denotes the first letter that we are reading. The second letter we're reading is on the top. Again, you see UCAG on top. And the third letter is on the right. Again, in each of the boxes, you have the UCAG in the third letter. So how do you read this genetic code? For example, if I want to know which amino acid corresponds to the code CUG, I'm going to start with the first letter, which is C, and I'm going to just switch over to the laser pointer. I'm going to start my first letter with C, second letter, which is U, so we are actually looking at this box here, and the third letter is G. So for CUG code, we will know that from here, it's actually coding for leucine. Similarly, you can use this method to find the amino acid of whichever 3-letter code that you come across. Now there are only 61 codes in this table that are coding for amino acids. There are 3 special codons here: UAA, UAG, and UGA, which are stop codons. And you have to remember that stop codons are actually not actually do not correspond to any amino acid. Now, just now we mentioned that there are 2 amino acids which are only coded by 1 codon. The first one is methionine, which is coded by AUG, and AUG also is the start codon. The other codon that codes for only 1 amino acid is UGG, which codes only for the other way around. The other amino acid that's only encoded by 1 codon is tryptophan, which is coded by UGG. Okay, so to recap, the genetic code is degenerate and universal. In the genetic code, there are 4 types of bases in mRNA that will encode the genetic code. And these 4 types of mRNA in 3 nucleotide codons will be able to specify 64 different combinations. But out of them, they only code for 20 different amino acids. That is because more than 1 codon can specify the same amino acid. In other words, the genetic code is actually degenerated. And such codons are also called synonymous codons. And if you go back to the previous slide, you'll notice that for a lot of these synonymous codons, only the 3rd base of the codon is the one that's actually going to be varying. So for example, for glycine, you have 4 different codes that all code for glycine: GGU, GGC, GGA, and GGG. You notice that the first 2 codons are GG across all the different codes. Only the very last codon, the very last letter of the codon is actually different amongst these 4 codes. And this last base is therefore referred to as the wobble base. Okay. And of course, just someone mentioned that many different species use the same genetic code system. That's why we call it universal, but there are indeed some exceptions as well. Moving on, we are going to introduce to you the next concept called reading frames. In an mRNA, when the mRNA is being read and being translated into a protein, the ribosomes will actually try to start the translation process at the start codon. And during the translation process, the ribosome actually moves in 3 nucleotide sequences. So every time it incorporates a new amino acid, it's going to actually move down 3 nucleic acids. Therefore, you can actually consider the reading of the mRNA to be in 3-letter to be words of 3 letters. Okay, and the start codon AUG will define the reading frame of mRNA because that is the place where the reading of the mRNA actually starts. So in this example, there could be a lot of bases even before the highlighted AUG. However, because the translation process only starts at the AUG here, it is therefore defined as the start of the reading frame. Now, inside this reading frame, we can actually already determine the protein sequence or the amino acid sequence that can be actually translated by reading this mRNA in 3-letter words. So the first one, AUG, gets translated into methionine, CCU gets translated into proline, GCC translated into glycine, etc. However, if you were to remove the 3-nucleotide base CCU, this will actually lead to the deletion of the amino acid proline here. However, the rest of the amino acids in the same, that the rest of the amino acids which are in the polypeptide will still remain the same. So you still have the methionine is still here, the glycine is still here, glycine is still here, serine here, aspartic acid here, and glutamate is here. So, and you have deletions or any other mutation that happen to 3 nucleotides in a sequence, which happens to be just one word in the mRNA sequence. This would actually not cause any problem in the sequence of the amino acids that's getting translated, because the reading frame, it doesn't actually changing. It still remains within the same reading frame that's starting from the AUG. And you can see that the rest of the letters still follow the same sequence as the original version. However, if there's only one nucleotide getting removed, in this example we're removing the 7th nucleotide G. If that nucleotide is being removed, the reading frame will therefore be shifted, because the next 3 letters that the ribosome is actually reading, instead of reading a GGC, it's going to read a GCG, and so on. And therefore, the amino acid sequence that this particular sequence is going to be translating things into will actually be different. Now, I want you to take a couple of minutes to refer back to the list of amino acids, and I want you to try to actually fill in the blank and tell me what's actually all these amino acids which are in the blank. Okay, let's see. The next code in this case is actually GCG, which corresponds to alanine. Moving on, GUA, valine, GCG, alanine, ACC, serine, and we don't really know what's happening after the G here. Okay, and you will notice that this sequence becomes very different from the other sequence that we had in the previous slide. So this kind of mutation that deletes or inserts 1 or 2 nucleotides, which will actually lead to a shift in how the RNA sequence is being read, is actually called a frame shift mutation. Whereas the deletion or insertion of 3 nucleotides, which doesn't cause the mutation to which doesn't actually cause the reading frame to actually shift, will then not be called a frame shift mutation. There are different kinds of mutations which can happen to the which can happen in the genes. Besides frame shift mutations, we have missense and nonsense mutations. They are not covered in this particular subject, but I just want to highlight to you that missense mutations and nonsense mutations are pretty much mutations that don't really involve shift in reading frames. Okay, so missense mutation is where the mutation probably changes the one of the amino acids to a different one, whereas a nonsense mutation changes an amino acid to the stop codon. Okay, but neither of these two kinds of mutation actually cause the reading frame to shift. So frame shift mutation is a special kind of mutation where 1 or 2 nucleotides are removed, which lead to the change in the reading frame. Okay, from the the genetic code list, you will also see 3 different stop codons: UAA, UAG, and UGA. And as I mentioned earlier, these stop codons don't actually code for any amino acid. They function to actually signal to the ribosomes and basically to the translation machinery that this is the end of the polypeptide synthesis, and they should when they reach this place, release the polypeptide that has been synthesized. So stop codon is also called the nonsense codon, and that's what gave rise to the name nonsense mutation, where one of the amino acids changed into a stop codon. The different stop codons are actually recognized by different types of release factors. The release factor is a molecule that has a similar structure to tRNA, but doesn't actually carry a amino acid with it. So these factors will recognize the stop codon, enter into the ribosome, and they will actually stop the translation and release the polypeptide as well as the ribosomes from the mRNA. Generally speaking, a polypeptide is a few hundred amino acids, but they are the length can vary depending on the type of molecule that is actually generated. Okay, after talking about the genetic code, which is the language or the rules how the message, the genetic information on the mRNA is translated into proteins. Next, we're going to look at different kinds of RNAs which are involved in the translation process. We covered this briefly in topic 2, but here we're going to look a little bit more into details what all these different kinds of RNAs do and how they actually function. RNAs are a very important kind of molecules in any living organism because they are involved in pretty much every step of the translation process. And all the different RNAs are actually produced by transcription. So you can imagine the transcription machinery manufacturing all the different kinds of RNA which are responsible for the actual production of proteins. Let's start with tRNA or transfer RNA. Now the tRNA is structured like a stem. Okay. And on one of the loops of the tRNA, that it actually contains a anticodon loop. So what happens is that the tRNA is going to actually try to recognize and bind to the codon on the mRNA. So when the ribosome is assembled on the mRNA, the tRNA is going to come in into the ribosome and try to base pair with the mRNA that the ribosome is reading. Okay. Now the tRNA molecules, they usually carry a specific amino acid which corresponds to the anticodon that is on the tRNA molecule. So a tRNA molecule is going to go into the ribosome, try to base pair with the codon that is in one of the sites on the ribosome. And if they are able to actually base pair perfectly, the tRNA is going to leave the amino acid to the ribosome, and then it will leave the ribosome later on. So tRNA basically acts as a transfer mechanism to carry amino acids, to carry the correct amino acids to the ribosomes to be assembled into proteins. Now tRNA, there are many kinds of tRNA. There are actually 61 different kinds of tRNAs. Every one of them corresponds to one particular codon. Okay. But we call them according to the amino acid that it carries on instead. So a tRNA gly is a tRNA that carries glycine. A tRNA lu is one that carries leucine, etc. So there are, of course, different kinds of tRNA, given that every anticodon, you'll require one different tRNA for every anticodon. So there are many different tRNA genes which are in our genome. So this is the structure of a tRNA, as you can see, starting from the 5' end, okay, which is which is where we have the phosphate group. We have from here a anticodon sequence at the bottom, right? So this is actually looking quite like a clover leaf with 3 different leaves here. So the leaf on the left and right are less important, so we're not going to talk about them. We're going to talk first at the anticodon loop, which is at the bottom of it. Okay, so this anticodon loop is actually complementary to the codon mRNA. So from the 5 prime to from the 3 prime to 5 prime, we have GGC on this anticodon, and therefore on the 5 prime to 3 prime on the mRNA, this anticodon should actually be correspond to CCG on the mRNA. Okay. And moving on, let's go all the way to the 3 prime end of the tRNA, and you have a little bit of a single-stranded region on the tRNA, which is called the acceptor stem. So this is the place where amino acid is actually bound to onto the tRNA. So for this particular tRNA that we have here, that has the anticodon GGC, which corresponds to the codon CCG, it should actually be carrying a proline on the acceptor stem. And when this tRNA goes into the ribosome and allowing the anticodon to actually base pair with the mRNA, make sure that it's actually the correct base pair, the tRNA will actually release the amino acid that's actually loaded onto the 3 prime single-stranded region, the acceptor stem, and this will be loaded onto the growing polypeptide chain that is attached to the ribosome. Once tRNA is once an amino acid is attached to a tRNA, this entire structure of tRNA amino acid, so whole complex, is called a charged tRNA. And the synthesis of charged tRNA is done using amino acid tRNA synthase. And for every different amino acid, there is one amino acid tRNA synthase, which is there to synthesize the tRNA molecule, the charged tRNA molecules. So again, I have mentioned this just now. For this particular tRNA that we have here, it will actually base pair with the mRNA sequence GGC, CCG in the 5 prime to 3 prime direction, and it will actually be loaded with a proline. Next important RNA in the translation process is actually ribosome. A ribosome itself is actually a complex of RNA and proteins, but the RNA is actually the catalytic center of ribosomes. Therefore, it's one of the most important things that's in the ribosome. So ribosome has RNAs, rRNAs, ribosome RNAs, and a lot of proteins. And in terms of structures, the prokaryotic ribosome is actually very different from the eukaryotic ones. The composition of the ribosomes, including which ribosomal proteins are there, how many are there, and the sizes of the ribosomal RNAs are all very different between prokaryotes and eukaryotes. In bacteria, there is one type of ribosome which is present in the cytoplasm, and it's called 70S ribosome. The 70S ribosome is made up of 2 different subunits. The small subunit, which is called the 30S, is again made up of the 16S rRNA together with 21 different proteins. On the large subunit, the 50S subunit, we have 2 pieces of RNA, 23S rRNA as well as 5S rRNA, and with it 34 different proteins, which have to come together to form the large ribosomal subunit. So you can see from this picture, it's actually a very, very complicated structure. The 30S contains the 16S RNA, which is the yellow thing here, together with 21 different proteins. They come together for this structure. 50S, made up of two pieces of RNA, 23S and 5S, together with 34 proteins, come together to form 50S RNA, or 50S, or the large ribosomal subunit. And the 50S and 30S will come together to form a complete 70S ribosome. On the other hand, in eukaryotic cells, there are many different kinds of ribosomes, which present in different locations in the cell. Of course, we're talking about the cytosol most of the time because that's probably where the most of the protein synthesis is actually taking place. We have also learned previously that there are ribosomes that are present in mitochondria as well as in chloroplasts because they have their own sets of DNA and their own sets of protein synthesis mechanisms. Okay, but we won't be talking about them in this topic. For the eukaryotic cytosolic ribosome, it is the ATS ribosome, which is this big orange guy here. Now, the ATS ribosome is again made up of two different subunits, the 40S, which is the small subunit, and the 60S, which is the large subunit. The small 40S subunit is made up of one piece of RNA, 18S rRNA, together with 33 proteins. And the 60S, the large subunit, is made up of three different ribosomal RNAs, 5S, 28S, and 5.8S, and 49 different proteins. And again, they have to assemble first into the small and large subunits, and then they will assemble together into the ATS subunit at the initiation of translation. Do you still remember which RNA polymerases are responsible for the transcription of these four different rRNAs? For 18S, 28S, as well as 5.8S rRNA, they are transcribed by RNA polymerase 1, whereas for 5S rRNA, it is transcribed by RNA polymerase 3. Now, ribosomes are actually a very interesting kind of molecule, especially ribosomal RNA, because different species actually have slightly different sequences in their ribosomal RNA, and because ribosomal RNA is probably one of the most common, most abundant kind of RNA which are present in cells, they are very easy to extract from the cells and very easy to analyze. And therefore, ribosomal analysis is actually used in species identification. So in this particular example, you guys probably have known this company already, Yuan Sang. They actually use the 5.8S ribosomal RNA sequence to actually confirm the identity of the polyceps that they use in their products. Okay, so if they are able to say that the rRNA sequence, or in this case rDNA sequence, is the same as the species that are comparing it to, they have a very high confidence that they are actually using the correct species for their product. On the other hand, you guys probably have heard of this. This happened quite a number of years ago when horse meat was actually found in Tesco burgers and actually a lot of mincemeat products, not just in the UK but in all over Europe. This happened probably about 6-7 years ago, about 10 years ago. Wow, that was a long time ago. Okay, so what happened was that people actually used this, the sequence of the 12S small subunit of the mitochondrial ribosome to identify the species where a particular piece of meat is coming from. Okay, so and from there they actually identified horse meat in the meatballs and the burgers that people are actually buying from supermarkets. This actually included IKEA as well. So for a brief period of time, IKEA meatballs were actually out of stock in Singapore because they actually have to import, or rather we have to export all these meatballs to Europe so that they have um, meatball that are not made of horse meat to consume. Okay. And this technology is also used in halal testing to see whether pork, whether there's any pork DNA present in the meat products, and therefore there may be contamination with pork. So ribosomal analysis is actually a very popular tool in species identification nowadays. Moving on, let's take a look at the functional size of the ribosome. And in this case, we are actually looking at the prokaryotic ribosome. So we have the 50S and the 30S subunits. Okay. So the shape of the ribosome is mainly determined by the structure of ribosomal RNA, which is basically the majority of the mass of the ribosome anyway. Now, if you were to actually cut this ribosome open from the middle, you will actually see the three different functional sites. We have from the right, the A site or amino acid site, the P site, the peptidyl site, and lastly the E site, the exit site. Now the reason why I'm going from the right to left is because on the right A site is the place where tRNA actually enters the RNA, and the E site is where the tRNA actually exits the ribosome. So this is basically coming when you go from right to left, we are moving in the same direction as the transfer RNA, the tRNA, that's going to be in the ribosomes. Moving on, let's take a look at the functional sites in the ribosome. The overall shape of the ribosome, what we're doing here, is mainly determined by the structure of RNA, ribosomal RNA, which consists of the majority mass of the ribosome. The one that we're looking at is a bacterial ribosome. We have the 50S subunit, which is the large one, and the 30S subunit, which is the smaller subunit. If we were to make a cross section of this ribosome, cut it open, we will actually see three sort of hollow areas inside the ribosome. The first one, A site, is on the right, P site in the middle, and E site on the left. A site, or amino acid site, is the place where tRNA actually enters the ribosome. The P site, peptidyl site, which is actually connected to the peptide exit hole. This is the place where the amino acids are added to the polypeptide, and also where the polypeptide is actually growing out of the ribosome. And lastly, the E site, the exit site, is where the spent tRNA gets ejected from the ribosome. So all these three sites are located inside the ribosomes. The last RNA molecule we're going to be looking at is mRNA, the messenger RNA, which contains the genetic sequence to be translated into the protein sequence. As we learned in part 1, prokaryotes have a polycistronic mRNA, which means there are multiple genes that are present on one piece of mRNA. On the other hand, for eukaryotes, the mRNAs are monocistronic, so there is only one gene on each of the mRNA molecules. The codons that we're looking at the genetic code are actually found on the mRNA molecules. And the ribosomes have to bind to the mRNA molecules to initiate the translation process. And as the ribosome move along the mRNA, tRNAs are going to come into the ribosome in order to for the amino acids to be added to the protein, the peptide chain. But the tRNAs and the codon have to match the codon on the mRNA in order for the tRNA's amino acid to be transferred onto the growing polypeptide chain. Okay, next, let's look at the translation process. Similar to transcription, there are also 3 stages in the translation process. We start with initiation, where the mRNA, the tRNA for initiation, as well as the ribosome subunits will actually come together to form a complex. Moving on, we have elongation, which is where the ribosome is going to travel along the mRNA in the 5' to 3' direction, and in the process, tRNA is going to come in, the correct tRNA is going to come into the ribosome and have their amino acids added to the growing polypeptide chain. And this is going to go on all the way until the mRNA encounters the stop codon, and that's where the different components will disassemble. So we have the ribosome actually dissociating from the large and small subunits are going to dissociate. The completed polypeptide going to leave the ribosomes, and all the different tRNAs are also going to leave the ribosomes as well. Step 1, initiation. Now the initiation is the place where the translation process actually starts, and the site where the translation actually starts on the mRNA is going to be very crucial, because that site, the particular site, is going to set the reading frame for the message encoded by the messenger RNA, Okay, so what happens, generally speaking, is that at the initiation process, the ribosomes are going to have to start they're going to actually look for the start codon on the mRNA, and only when the start codon is found will the ribosome actually start to initiate the process. Remember, start codon is AUG, so the AUG codon is going to bind to a special tRNA, which is called initiation tRNA, and this tRNA will have a anticodon which says UAC, right? And this start codon is going to bind onto the mRNA and carries with it a methionine. For the prokaryotic cells, there is no 5' cap to signal to the ribosome where to start searching for the start of the translation. So the ribosome is going to try to locate the start codon. So how does it do that? In prokaryotes, the mRNA actually contains a very specific ribosome binding site called the Shine-Dalgarno sequence. And this Shine-Dalgarno sequence is a couple of nucleotides upstream of the AUG codon, the start codon. And the Shine-Dalgarno sequence is going to form base pairs with a small subunit, small ribosome subunit, because the subunit also contains some RNAs, right? And with the Shine-Dalgarno sequence, as well as the MET initiation tRNA, it's going to try to actually position the small ribosomal subunit at the start codon at the AUG codon. And once the small subunit actually sits at the correct place, then the large subunit is going to come in to assemble onto the small subunit, and that will complete the entire ribosome. Now, for prokaryotic cells, remember that the mRNAs are actually polycistronic. So you can imagine there being multiple Shine-Dalgarno sequences along the entire mRNA, one before each of the genes that piece of mRNA is actually carrying. So you have Shine-Dalgarno sequence, followed by a start codon of the first gene, stop codon, and a little bit later, the next Shine-Dalgarno sequence for the next gene, start codon, stop codon, and the next Shine-Dalgarno sequence for the third gene, stop codon, etc., along the entire mRNA molecules. So you can actually expect multiple initiation that takes place on one piece of mRNA as well. Now let's break this down step by step. At step 1, we have the source subunit binding to the mRNA. So we have this yellow piece of mRNA molecule here. And the strand diagonal sequence, or in this picture, is actually labeled as ribosomal binding sequence. They are basically the same idea. It's a little bit upstream of the start codon, in this case the green color nucleotides here. And this strand diagonal sequence is complementary to a short sequence on the 16S RNA, which is inside the small ribosomal subunit. And this will actually base pair with each other because they are ribosomes. I mean, they are RNAs. They are single-stranded, right? So this will actually cause some sort of hydrogen bonding between the mRNA and the 30S small subunit. And after that, once the binding of the ribosomal subunit and the mRNA is done, next we have the initiator tRNA, okay, which carries a methionine. In this case, it's actually formylmethionine, the initiator tRNA. It's going to come in and bind to the start codon. And now, if you kind of still remember the three locations inside the ribosome, A, P, and E site, you'll see that the initiator tRNA is actually bound to the P site of the small ribosomal subunit. Now, the initiator tRNA carries with it a formylmethionine, which is a slightly modified version of methionine, but the formal group can actually be removed after the translation, so that at the end, the first amino acid may not actually carry the formyl group. Moving on, after the binding of the small subunit to the mRNA, as well as the binding of the first tRNA to the start codon, next is the binding of the large ribosomal subunit. So basically, you see this big 50S ribosomal subunit that comes in, and this is gonna position also the tRNA, the first tRNA, inside the P site. Okay. And at this moment, we have an empty E site and we have an empty A site. And then it's the end of the initiation stage. After this stage, the ribosome is gonna move along the mRNA, okay, and then new amino acids, new tRNAs are gonna come into A site and try to base pair with whatever new sequence that appears in the P site. Now let's take a look at eukaryotic cells instead. For eukaryotic cells, the translation initiation process is actually a little bit more complicated because it involves more molecules, and it also involves a lot of reading by the ribosomes. So in eukaryotic cells, the initiator tRNA, which is coupled to the methionine, is going to be loaded onto the small ribosomal subunit first, together with a lot of molecules, a lot of proteins called eukaryotic initiator factors, EIFs. The small ribosomal subunit with the tRNA is going to recognize the 5' cap, and it's going to bind to the 5' cap of the mRNA. And then once it finishes the binding, it's going to move along the mRNA in the 5' to 3' direction. It's going to search for the start codon AUG. Of course, there are also a lot of other molecules which help with the process as well. Once the ribosome reaches the first AUG, the eukaryotic initiation factors, the EIFs, are going to dissociate from the small subunit, and then this will allow the large subunit to actually come in to assemble at the start codon, and therefore initiate the translation process. Now let's again take a look at how this actually takes place. So at the beginning, we have a piece of mRNA down here, the orange color molecule here, and we have a 5' cap on the mRNA. Now remember, this was added inside the cell nucleus during the post-transcriptional modification. And then we have the small ribosomal subunit, which is associated with the initiated tRNA, as well as the eukaryotic initiation factors. All these molecules are going to bind to the mRNA after the 5' cap. Okay, and this entire complex is going to go, it's going to scan along the mRNA until it reaches the AUG, the start codon. When that happens, the EIFs, the eukaryotic initiation factors, they are going to actually dissociate from the initiation complex, and the large ribosomal subunit is going to come in to bind to the small subunit, and therefore forming a complete ribosome. And this will complete the initiation process in eukaryotes. Step 2 of translation is, of course, elongation. Now, this process is basically the synthesis of the polypeptide chain while the ribosome is reading the mRNA. So the process begins with the charged tRNA, which carries a new amino acid, and it will actually come into the ribosome. And then it will actually be only if the charged tRNA has the correct anticodon, then you will actually be able to stay inside the ribosome and have its amino acid being incorporated into the protein sequence. It's a fairly fast process, 15 to 18 amino acids per second in prokaryotes and about 6 amino acids per second in eukaryotes. Now, the elongation process is again facilitated by a lot of other molecules, EF2 and EFG, etc. And of course, there will have to be a lot of energy being used because there's going to be a lot of synthesis of peptide bonds. And the energies are actually supplied by hydrolysis of GTPs. The elongation process is again a 3-step process which repeats again and again in order to complete the synthesis of the polypeptide chain. The process is actually quite similar between prokaryotes and eukaryotes, so we're going to be using prokaryotes in this case, for example. So step 1 is the entrance of a charged tRNA that will carry a new amino acid into the A site. Okay. So the tRNA is going to come in, and together with it, it has the anticodon sequence. And you see in this particular tRNA, the anticodon sequence is GCA. Okay, and the codon sequence that it will be able to bind to will actually be CGU. And if you look down here, the next 3 nucleotides here are, well, CGU. So in other words, this tRNA that comes into the ribosome is actually going to be the correct tRNA. Okay, so it's going to come in and it's going to bind to the mRNA because there is going to be hydrogen bonding between the anticodon and the codon sequence. Okay, and the elongation factor, EF2, is going to provide some energy, hydrolyze GTP to provide some energy, and this will help to bind the tRNA to the A site. Once the tRNA comes into the A site, and make sure that it's actually the correct tRNA that has to be there, okay, by hydrogen bonding. The next step that's going to happen is the peptidyl transfer reaction. So remember in the previous slide, this chain of amino acids was actually bound onto this tRNA on the left, right? So what happens is once the next tRNA comes in and verified that it's correct tRNA, there's going to be a peptidyl transfer reaction that's catalyzed by the peptidyltransferase. The peptidyltransferase is actually the function of the ribosomal. This is the function of the ribosomal RNA. Okay, so this is actually not a protein enzyme is actually an RNA enzyme. So this RNA is going to actually transfer this entire peptide chain from here onto the next amino acids on tRNA. So in other words, the polypeptide is actually transferred from the P site to A site. And then once it's done that, it will actually form a new peptide bond between the peptide chain and the amino acids. So the amino acids then incorporated into the polypeptide chain. Now the next step is actually a translocation of ribosome. So what happens is that the ribosome itself, once the amino acid has been correctly transformed, is going to move along the mRNA by 3 nucleotides. So again, facilitated by EFG, which will actually move the ribosome to the next codon position. Now, during this process, the tRNAs, because they are actually hydrogen bonded to the mRNA, the tRNA will actually remain at their original sites, while the ribosome itself moves ahead. So the first tRNA that we saw in step 1, which is this one that has the anticodon AGC, is then pushed into the E site, and E site has a hole that will allow the tRNA to exit. The second tRNA that we saw coming in is then moved into the A site together with the growing polypeptide chain, and then to the P site. So and then the A site is now empty. This will actually allow the next tRNA to enter, and then it will basically go back to step 1 of the elongation process. Okay, so the 3 steps of elongation. The first step is the entrance of tRNA into the A site. The second step is the formation of peptide bond by the peptidyl-transferase reaction. The third step is the translocation of the ribosome and the release of the spent tRNA, which will also allow the entrance of the next tRNA. The last step in translation is termination. The termination process takes place when the stop codon is reached on the mRNA. There are 3 different stop codons: UAA, UAG, and UGA. Now this is where things start to get very interesting, because for prokaryotes we have different kinds of releasing vectors: RF1, RF2, and RF3. On the other hand, in eukaryotic cells we have only one kind of release vector, ERF. So regardless of the kind of releasing vector that we're talking about, they all mimic the structure of a tRNA, and therefore they can actually enter the A site of the ribosome. And once they enter, we go through a 3-step process to stop the translation process. And step 1 of the termination is the binding of the releasing vector at the A site. So now this is going to be a very similar picture to what we have saw previously. We have the mRNA here, we have a tRNA in the P site, and we have a polypeptide chain that goes out of the site of the ribosome and is attached to the tRNA. Now in this case, we have the ribosome now encounters a stop codon, so one of the 3 different stop codons. And when that happens, we have a release vector, a protein, that will actually enter the A site of the ribosome. It will bind to the stop codon. And after the binding of the releasing vector to the stop codon, this will actually trigger the ribosome to release the polypeptide chain as well as the tRNA. So the tRNA is going to be translocated into the E site and get ejected. The polypeptide chain is going to actually exit the ribosome from the hole on the site. Now, after getting rid of the tRNA as well as the polypeptide chain that's being generated, The last step in the termination process is of course the dissociation of the ribosome subunits. The two subunits are going to dissociate, and this will also release the mRNA as well as a releasing factor from the complex. So in this way, we can terminate the translation process. Again, the termination is a very similar process between prokaryotes and eukaryotes. So what differences between prokaryotes and eukaryotes during translation have we discussed so far? So, well, let's talk about some similarities first. Both prokaryotes and eukaryotes have the same three stages of translation process, and most of the differences actually occur in the initiation stage, where different factors are going to be needed. Okay, so for eukaryotic cells, we have EIF, the eukaryotic initiation factors involved in the process. Okay, and for tRNA is tRNA met instead of the tRNA formylmethylene, and the mRNA actually binds to the 18S ribosomal RNA in the 40S ribosomal subunit. Okay, the next difference, which is probably the most important difference between prokaryotes and eukaryotes, is at the binding of the ribosome to the mRNA. For prokaryotes, the ribosome actually binds to the strand diagonal sequence, which is a little bit upstream of the start codon of the mRNA. Whereas in eukaryotes, the binding of the ribosome is actually near the 5' cap. So the 5' cap is first recognized by the cap binding proteins, and then those cap binding proteins are going to help to actually locate the small subunit onto the mRNAs. So that's one of the major differences at initiation between prokaryotes and eukaryotes. And of course, we have the Kozak sequence, which will guide the location of the start codon AUG downstream of the 5' cap in the mRNA sequence. Okay, so in this particular example we have here, the AUG is in the middle. So this is the start codon. And we have a Kozak sequence, which is basically at -3 and +4 locations. This will actually help to guide the translation machinery to the correct position of the AUG start codon. So in most of eukaryotic mRNA transcripts, we can find this Kozak sequence present. This is actually a very important initiation site for eukaryotic mRNA for translation in eukaryotic cells. And it's actually conserved for a lot of the eukaryotic genes. Now, the presence of the Kozak sequence is so that the ribosome can actually recognize the AUG correctly and to be put into the correct reading frame. Because there may be multiple AUGs along the entire mRNA, so this Kozak sequence will help to position the ribosome at the correct AUG to actually start the translation process. Now, one last thing that I want to talk about is how the transcription and translation can actually be coupled in bacteria because of the lack of the nuclear membrane. So in bacterial cells, once it actually starts to transcribe the gene, before the transcription actually ends, you can actually already have translation that takes place on the piece of mRNA that's actually being produced because they actually happen at the same location, the same space. Okay, so because of that, we can actually expect prokaryotic cells to generate protein a lot faster than eukaryotic cells, because once they start transcription, they can actually start translation already. Okay, so there's no lag in time between the two processes, so it makes prokaryotic gene expression a lot more efficient, which is kind of important for prokaryotic cells because they actually need to adapt very quickly to the environments they're in, and therefore they may actually require the expression of the genes a lot faster than eukaryotic cells. And that is all the content in part 2 of the lecture on translation.